Welcome, everyone. This is Derek Pizarro again, AST Environmental. I have my colleague Mike Mazarese here with me today. I'll be emceeing his presentation, Chlorinated Solvent, Daughter Product Management, and Expedited Remediation. This will be a short format webinar, so we're probably going to try and keep this right around 30 minutes and some Q&A. Myself and Mike been with AST for a while. Mike will introduce himself. He's been in this business doing environmental remediation over 20 years. He's a Penn State grad, BS, MS in environmental engineering. One thing to note here, this product does focus on RPI's CAT 100. There are some tie-ins to some other products at Remediation Products and Group, but I would just like to, for our new friends and colleagues joining us here today, make clear that RPI and AST, while we have a partnership, we are two separate entities. And AST is the global design and installation partner for the group. So with that said, Mike, please take it away.
Thanks, Derek. Thanks everyone for joining us this morning or this afternoon, depending on where you're joining us from. As Derek mentioned, I'm a senior engineer with AST. I've been here for 9 years and been in the in situ remediation industry for close to 23. And I'm based in the Denver, Colorado area. And so the focus of today's talk, as you can see, we're going to be discussing daughter products and sort of managing them from in a couple different ways, right? So there's our sites that we get involved with, and you all as well, where we're dealing primarily with parent products, so PC, TC, for example, and then there are sites that already have had some conversion, whether that's natural or whether it's by the addition of an electron donor, and we'll talk about each of those situations. And so I'm going to make the assumption that everyone here is familiar with these processes, right? And so I'm not going to show the graph that you see in every presentation of PCE going down to ethylene. We all know daughter products are going to be generated if you have an electron donor, whether that's some natural organic carbon, for example, or if you're adding an electron donor, so something like lactate or something like an emulsified vegetable oil, and you're going to generate daughter products. And we have sites that you get complete conversion, and some sites that you don't. And, we're going to talk about on the next slide, some reasons why we do get that stall at, say, DCE or vinyl chloride. And the issue, excuse me, is that when you're dealing with source areas, which is kind of the focus of this presentation, you're dealing with, single, double digit, say, milligram per liter concentrations of parent compounds. You're going to get similar concentrations generated of your daughter products. I mean, I've seen sites, I've reviewed sites that have, 10 milligrams per liter of vinyl chloride rolling off site. And so that's obviously something that we're trying to avoid, and we'll focus on that the rest of the presentation. And what can we do to try and minimize that vinyl chloride generation, and what can we do as sort of a fail-safe on the downgradient edge to ensure that we don't have these, DC vinyl chloride that don't sorb the soil as well as, say, TCE or PCE. How do we stop them from leaving the site or impacting a sensitive receptor? So, and there are multiple reasons why you're going to have daughter products generated and have them persist. One, it could be a geochemistry issue, depending on the DO, concentration, depending on your ORP. So if you've got a couple milligrams per liter of DO, you've got positive ORP. A lot of those sites, we don't see any daughter products generated at all, or minimal quantities. Could also be a pH issue, right? So DHC, has a pretty tight range of pretty tight pH range where they're gonna be happy and viable. Microbiology in general, I think it's good to have do some baseline microbiologic microbiologic assessment work to see what's the current, what are the baseline conditions out on your site? Do you have DHC present, for example? Do you have functional genes present? Lack of sufficient organic carbon. So this can be natural or injected. So if you don't have enough, if you have some organic carbon, maybe you get some conversion naturally, then that's going to stall out once that's depleted. Or if you just don't add enough electron donor, or it runs out before you get full conversion. But I also think it's important. The next bullet kind of ties in is distribution. So if you're going in and injecting a electron donor, for example, and you get great distribution in one area or in one vertical interval, one specific vertical interval, well, you're going to get DCE vinyl chloride generation there. But if that now flows into an area down gradient where you didn't get good distribution, that's going to keep moving through, right? So distribution is important for any amendment, but is germane for this conversation as well. I think incomplete CSM, we'll talk about high density sampling as part of this discussion as well today. And I think that's really understanding, and I think that microbiology ties in here as well as part of that CSM. So understanding from a saturated soil standpoint. So again, focusing on source areas, any source area, saturated soil is going to be super critical. Understanding how those concentrations vary with depth. And when you're dealing specifically with parent and daughter products, I'll show you some data here on our third site where ERD was done, and you could see where it had an impact vertically, and you can see where it didn't, where we got conversion, where some conversion was seen in some intervals, but not in others. So it's really important to be able to know how much saturated soil mass, for example, is going to be bleeding into your area over time, because it's going to help manage expectations. And then lastly, for design/capture, what I mean there is making sure that ties in with my comment before about having a down gradient capture technology that's not ERD based, so something that's abiotic, to be able to capture any daughter products that do ultimately flow out of these source areas. So we're going to talk about three sites, and they're all quite different, but there are some similarities. These are all source areas. There's no PRBs in the source area. So there are PRBs on each of these sites on the down gradient side, and I'll touch base on that here in the last bullet. It's all unconsolidated, relatively shallow. Everything's less than 50 feet. It was all done with direct push. Each of these sites had an RDC performed. So RDC is remedial design characterization, and it's just high density soil and groundwater sampling to help firm up and give us a more accurate conceptual site model. Most of these sites had some level of microbiological assessment work done. The third site that I'll show actually collected samples over time, over the course of the project for about 5 years. And then lastly, each of these sites had a BOS 100. So BOS 100 is an abiotic degradation pathway. So it's activated carbon that's impregnated with reactive iron, and as a down gradient control to help capture anything that does ultimately flow out of these areas. And I think that's very important. And I think for all ERD technologies, I think we want to make sure we've got something down there to be able to capture, because again, regardless of what you're doing from an ERD standpoint, it's never going to be 100% complete. You're always going to have some, say, cis vinyl bleeding out of these areas, especially when we're talking about milligram per liter type starting concentrations. We want to make sure we've got something down there as a fail-safe. Okay, so I don't focus on technology here much. Those have been done in other presentations. Dr. Ed Winter with RPI did a presentation a few weeks ago that specifically talked about CAT 100 and daughter products. So I encourage you to check that out. That's in our webinar archive. And I'll give you a link to that here at the end of the presentation. So the core for CAT 100 that Derek had mentioned is BOS 100. So that's the activated carbon that's impregnated with the iron. So that by itself gives you very similar chemistry to zero valent iron. It's important to note, I think, that it's not a mixture of just carbon and iron. That would give you, because if you think about it, so the way that BOS 100 works, you get your PCE, for example, adsorbing into the pore structure of the activated carbon. It's interacting with the iron to get degraded via beta elimination. If you have activated carbon and iron just physically mixed together, now you've got your PCE adsorbed into your activated carbon and does not have contact with the zero valent iron. So those are quite different technologies, even though they have similar components. So with the BOS 100, we add an electron donor. So that can be either something like a food grade starch or chitin. We've used both. And there's a couple sets of bacterial cultures that are also applied. One set is specific to the CVOCs, and another set is designed to help break down those carbohydrates. And lastly, there's a nutrient package as well. So that's all slurried together, and it's all injected as one product as a slurry using direct push in these examples. So we're going to talk about 3 sites, as I mentioned, and I tried to pick sites that gave us kind of different results based on the baseline conditions. So number 1, site with elevated parent compounds, very few daughter products to start with. This would be your site where you've got really no natural degradation going on. It's mostly 90%, say, plus parent material. Second site is the flip side of that, where we've got some natural degradation or some ERD work done, where you've got at least some conversion, where you've got some DCE vinyl chloride just at to start with at baseline. And then number 3 is a site that had significant saturated soil mass, and specifically significant daughter product saturated soil mass. And so the signatures that we'll see in the data for all 3 of these are quite different. So the first site that we'll talk about was part of a phased CAT 100. You'll see there in that bluish, purplish square where you see MW18. It was part of a combined remedy where there was a large ISCO soil mixing. That's the red area that was permanganate, sodium permanganate. And there was a PRB installed downgradient with BOS 100. You see that in green. The CAT 100 work was done a little more than 3 years ago. And you can see the specs there, about 7,000 pounds, 70 injection points with an injection zone of 8 to 30 feet BGS. So I took this data and I broke it to give us kind of a nice little time lapse here. So this is our starting data points. So you'll see total CVOCs there in yellow, and just below it, you see TCE. So we had almost 100% of our CVOC mass was in the form of TCE. The vertical line represents when our injections were complete, calling that day 0. And about day 150, call it about a half a year into the project, I would consider this sort of a response that you would expect on another ERD site. You could probably, the TCE did degrade very, very quickly, which is something that we see on other CAT sites as well. Even at close to 50 milligrams per liter, that was non-detect at 150 days. And you can see that it's a slow decrease there. So it's not just adsorption onto the activated carbon. But we do see a concurrent spike in DCE, so that's in orange, up to over north of 70 milligrams per liter. And then we see our vinyl chloride increase as well, somewhere, call it 12 milligrams per liter. We fast forward to about a year post-injection, and we see all the concentrations have come down significantly. Total is below 10 milligrams per liter, but we still see about 5 milligrams per liter or so of DCE and about half of that of vinyl chloride. Move out another year, and we see all the concentrations have come down. I'm going to show you a data table here in a second, because I know with that scale, it's hard to see what those concentrations ultimately are. So I'll show you that here in a second. But about 2 years into this particular project, most of the parent and the daughter products were reduced significantly. And so about 3 years now, which is where we are, you can see we're non-detect for TCE, which we have been since early on. And we've reduced our DCE and vinyl chloride both below 10 parts per billion. And I would expect those concentrations to continue to decrease to get us down below the MCLs. And so you see here, so again, starting at about 48, the dealing with the TC PC is pretty easy. What is different for me, having worked on traditional ERD sites over the years, is really that DC generation is not unique. But how quickly that DCE and then the vinyl chloride did decrease, I think, is unique to this technology. And I would say, just as a sort of a common, just another comment that we see on other CAT 100 sites, generally don't see a stoichiometric production of vinyl chloride. This is what you see here is pretty typical, where we do see the DC increase, but the vinyl chloride is usually a lower concentration. We don't see an equal spike. Okay, so, and just one quick aside, most of these sites have standalone webinars that we've done that are in the archive or have been presented as posters or platforms at conferences. So there's more data available, there's more information. If you're interested in any of these, please let me know. So the second site is that inverse, right? So we've got on this site, as we'll see, there we were starting with more daughter products than we were parent products. And so here, this site was presented by my colleague Bill Brab recently, and he went through. We're in the middle of, or I guess we're at the end now. But this was about a 10-year phased approach, and this was also very, very similar to the first site. Had an ISCO component, so there was some soil mixing with sodium persulfate on this particular site. There was some BOS 100 applied. I'll show you the piece of the site where that downgradient PRB was. I'll show you that here in a minute. But the CAT 100 for this particular area was installed almost 3 years ago in September of 2020. So for this site, I thought I'd show kind of what the RDC looked like. So this site, fairly large site, had 44 total soil borings over a couple different phases, sampling soil approximately every 24 inches down through the saturated column. Also looked at shallow versus deep groundwater, and also sampled all the full monitoring wells. And then we look at obviously besides the volatiles, we're looking at all the anions and as well as all the dissolved gases. And this site had some microbiological testing done as well, which is not presented in this presentation. And all those samples came to the lab, the RPI lab, which is out here in Denver, where I'm located. And all that analysis was run pro bono by RPI. And that's for all of their projects. And actually, even if it doesn't ultimately end up being an RPI AST project, you don't get an invoice for that analysis. We feel like this is the data that we need to properly assess your site and to make sure we've got a good design, but then also we can properly manage expectations. So we're going to talk about two different areas. So this PDC 14, which is close to the source area, and PDC 3, which is down gradient, that's where the BOSS 100 was applied, and PDC 14 was where the CAT 100 was applied. So walk you through this. So on the primary axis, PDC 3, this is the down gradient area. This is the blue line. So we'll see here this is total ethenes. I'll break it out into parent daughters here in a minute. But the blue line is PDC 3 before and after the BOSS 100 PRB was installed. And this is one of our longest data sets for BOSS 100s. We've got over 9 years of data. We do have a blip here, and looks like late 2018, but it's generally been reduced and has stayed low for the duration of the project. The orange line represents what's coming into this area. So this is up gradient. This is the PDC 14. So this is where the CAT 100 was applied. And that's this solid vertical line that you see here on the right. So everything to the left these are all our up gradient baseline concentrations that are flowing into this PRB area. And so after the CAT 100 was installed, we see a nice downward trend here down to down again. This is total CVOCs, decreased down over the course of about 2 years or so. Now, to break that out, the reason why this particular site, I like to include this site and this particular well, is if you look at those baseline concentrations, so there was some natural ERD that was going on here. We see a significant conversion to DCE with some vinyl chloride generated baseline, and looking at 144 milligrams per liter of DCE to start with. And these samples were collected. This line represents when the injection started, and these samples were baseline, is what I'm trying to say. This line needs to be moved over. These weren't taken post-injection. But after we did the injection work, we see the concentration significantly decrease. We're at about a year into the project here. We're at non-detect for both PCE and TCE. So that's very similar to the first project that I showed, and others similar to other projects where pretty easily deal with those parent products. But on this site, what was surprising to us is, how well the CAT dealt with the DCE and the vinyl chloride, even at those elevated concentrations. And so we're currently about 2 years of data that we have, looking at, triple digit ppb, 123 and 231 for DCE and vinyl chloride. And I would suspect, similar to the first site that I showed, if we were able to grab a sample at year 3, we'd see the concentrations continue to decrease. So we'll cover one more project here, and then we'll go ahead and open up the Q&A. So this third site also had significant concentrations in source area, but also relative to the other sites had significant saturated soil mass. And I'll kind of walk you through that table down here at the bottom after I get through these last couple of bullets. So similar to the other sites, it's a multi-year phased approach, which is for our other sites, other large chlorinated sites, kind of sensing a trend there. We are working on these sites year after year, sometimes using different technologies or just attacking various different parts of the site. This is one of our first CAT injections, or one of our first large scale CAT injections. This was done in the first quarter of 2016. So we have a good, nice long data set. And so the RDC, this is an RDC point that was done slightly down gradient of the well that I'm going to show you. And as you so this is the data output that you get from us. I obviously isolated just these 3 analytes. But you can see starting at about 8 to 10, not a lot of mass. We start to pick things up in the 10 to 12, and these are all in micrograms per kilogram. And we see it primarily as TCE, and that trend continues down to 16 feet. But then we get into a zone here. Now, if you remember, I told you that ERD was done out on this site, and I would suspect that these are the zones where the product was adequately distributed. And so we see in 16 to 18, for example, complete conversion from TCE to cis and vinyl. And similar to 18 and 20, we see a larger percentage of the saturated mass being being one of these two daughter products. But then once we get down below 20 feet, it flips back, right? So now we've got significant concentrations in saturated soil, 20 to 24, and then it flips back here, 24 to 26. So I guess what I'm trying to say is it's really important to have this information, because now we can see, because just like with any technology, whether it's bio or whether it's ISCO or whatever you're using, we're treating groundwater, right? And we're driving mass off of soil into groundwater so that it could be subsequently treated. And so when you've got these levels of saturated soil mass, we've got to slowly bleed this out of the saturated soil, and it's going to take some time. And so when I show you the data for this particular well, it'll start to make sense, because there's a lot more noise than what we saw on the previous sites. So here's the facility, multiple degreasers. We're going to focus on this area right here, this green box, this blue box here. This is approximate boundaries of our treatment area and cross section. MW71 that I'm going to show you is right here. So here's a plan view of the site. Here's that degreaser, which is in the red square. MW71, very close to the source area, is the data that I'm going to show you. These are all of our injection points that extended outside the building. So down here, not shown, actually if I go to the left here, this is where the Boss 100 PRB was installed. Again, if you're interested in some more information on this site outside of what I'm presenting, reach out to me and I can get that to you. So had about a 5,400 square foot treatment area, about 11,000 pounds of CAT 100 and 215 injection points. And it sounds like a lot of injection points. I know it is, but this is relatively shallow groundwater. And when you're injecting slurries in relatively shallow intervals, we've got to go with relatively low shot volumes to be able to make sure that we're keeping our reagent within our design radius of influence. Because you start pushing that radius of influence, especially on the shallow site, we've got a lot of utilities. You're going to have a lot of daylighting. You're going to have the material following preferential pathways and not going where we need it to go, right? And so here's that data from MW71. So as you can see here, the one similarity is that we see the TCE concentration decrease immediately. That's similar to the other sites. But if you look at the DCE, which is in blue, and the vinyl chloride, which is in gray, we see a lot of noise. And we attribute that to all that saturated soil mass that we identified in that RDC phase. And it took about 3 years or so in general, 3 years to this point, give or take, for us to really bring these concentrations down. And then this is year 4, more or less, to where, the concentrations now have decreased down significantly. So key here is if that's the importance of that saturated soil data, because now we can manage expectations that, hey, it's not a 2-year project, or 3-year project, similar to what we saw in those other examples. This might be 5-year plus. Now, this was with one injection too, which is really important to note. And one thing that I plotted on this particular graph that I didn't show on the others is the ethylene generation over time. Now, that is on the secondary axis. So the scale was slightly different. So you can see here we've got a nice steady increase of ethylene over the 5 years that you see here of the monitoring data. I also I mentioned there was a site where they had done microbiological testing over the course of the project. Here's that data from MW71. And so dihalococoides here, we see that's on the far left. We see at about a 10 to the 2 concentration to start with. In general, we see some DHC. I mean, it could be usually it's a pretty low concentration. The bacterial blend that we add with cat does not contain DHC, but by the addition of the carbon and the electron donor and all of that, we get those concentrations into that 10 to the 4, 10 to the 5 sweet spot that we like to see. And we see the Dehalobacter desulfomonas concentrations generally increase as well. We see the vinyl chloride functional genes, which were non-detect at baseline. We saw those concentrations increase over time, as well as the total U bacteria. So, all right. So we're almost 30 minutes in. So I'll go ahead and wrap up, and we can open up the Q&A. So I think one thing that sets the stage for all of these sites is just that RDC process and understanding, where your saturated soil mass is vertically and what that breakdown looks like from a parent versus daughter product perspective. And it really helps us surgically apply the material aerially as well as vertically, and then really important piece of that is helping to manage client expectations. And so I think we've been able to show with, Cat 100 has been on the market long enough now that we can safely say that it, expeditiously, manages daughter products relative to ERD for sure. And lastly, and I know I've driven this home a couple times, if you've got an existing ERD site that's got some daughter products, and we work on, those sites too, where maybe you're injecting an emulsified oil upgradient, and we're just dealing with the downgradient PRB piece. But if you're doing any sort of ERD, whether it's CAT or it's another technology, you should really make sure that you've got something downgradient. We think it's best available technology for this purpose is something like, BOSS 100, using that abiotic degradation pathway to capture anything that's going to be moving out. So I guess take questions. Thank you for your time. There's my email. There's our website. If you are looking for project summaries, our webinar archive as well has a couple of these sites have their own standalone webinars, or at least are broken out into case studies. Please reach out. That's the QR code for my LinkedIn if you'd like to connect with me that way. And otherwise, thank you for your time. And Derek, if you want to come back on, and we can open up the Q&A.
Sure. Thanks, Mike. There is a Q&A box as part of the Zoom presentation. If that doesn't work for you, there's also a chat function. Feel free to enter any questions you haven't done so already in either of those two places, preferentially the Q&A box. Mike, I'd like to ask one question that I get asked a lot when I talk about Cat 100, because a lot of our really good long-term case studies focus on overburden sites or transition zone. Question I get usually is, has Cat 100 been used in bedrock or applied in bedrock?
It has. I've been involved in a project in California at an Air Force base where it's been applied. I would say the most of our work is going to be in the overburden or the transition zones. So those of you that have sat in on other webinars where we've highlighted our geotap or pre-drill technique. We've done a number of sites where we've, pre-bored with sonic or augers to get into weathered bedrock, and be able to, and because we're injecting slurries, we can pressure inject through the bentonite that we can place in that hole. So I would say more in transition zones than bedrock, but we've done more Boss 100 work in bedrock to date.
Right. Some questions coming in now. Thank you for them. Has AST or RPI ever looked at geochemical data to review groundwater data that poses a challenge for CAT 100 or Boss 100, such as low pH? Or how frequently do you run into geochemical conditions that are adverse for these products?
I would say, everything's obviously got limits. But I would say that when you've got the activated carbon piece in there, it allows us to create a microenvironment for the bacteria where they're not exposed to those pH. So I think you've got more wiggle room than you do with a standard, say, ERD, where you're trying to keep it, neutral, plus or minus a unit, maybe a unit and a half. I think you could probably get down into the 5 to 9 range is going to be reasonable for CAT 100. So Boss 100 is really going to be fairly independent of geochemical parameters, and things like, if you have a high dissolved oxygen, for example, it's something that because it's reduced iron, right? We've got to, we've got to account for that. So, for example, if you're doing a Boss 100 PRB, we need to account for the oxygen flux going through the system. So, there's going to be extremes, but I'd say you'd have to have a, pretty aerobic, fast moving aquifer for Boss 100. And then when you're talking about adding electron donor, that takes care of kind of your elevated positive ORP situation, because you're going to drive that down with your electron donor. So I would say pH, if you're, below 5, above 9, that's where we can, look at some sort of buffering perhaps. And then DO, it's kind of, we've done work on some pretty aerobic sites to date. And I think it's, again, because the bacteria are adsorbed onto the activated carbon, and then they're somewhat insulated from those, either low pH or high DO environments.
Great, and thank you for that question. Another one here: does CAT 100 work on commingled contaminant sites like CVOCs, petroleum hydrocarbons, and PFAS?
Well, PFAS is the easy one. RPI does not promote their technologies for on PFAS sites, because there isn't a there's no degradation mechanism, as we all know. It's just activated carbon. So I would say, it could potentially be used if everyone agrees that, hey, it's just adsorption, which seems like what most folks are doing these days from an in situ standpoint. But we haven't yet gone down that path. AST is working with Setco to inject their Fluorosorb technology. And so I would say if it was a commingled site that had PFAS, we would probably look at using a combination of, say, trap and treat technologies with something like Fluorosorb, that we feel like is a better adsorbent than activated carbon. To go back to the other commingled sites, so if you've got things besides chlorinated ethenes, so you've got ethanes, methanes, chlorobenzenes, for example, things like that, or even petroleum hydrocarbons, we've had sites with all of those, or a combination of them. And so it's a matter of, I think that RDC is really important. Maybe there's some bench work that needs to be done. Maybe there's some tailoring of the bacterial cultures that need to be applied. But we have had sites that have had pretty wide mix. And that's actually a good question and timely, because Dr. Ed Winter's next presentation, I think I don't know if it's going to be in August. It might be after the Labor Day holiday here in the States in September, is going to focus on mixed plume remediation, specifically with CAT 100.
Great. I'm going to try and combine two questions here into one for you, Mike. Can I use CAT 100 in a PRB? And then kind of the next piece of this is, do I have to use BOSS 100 as part of a CAT 100 PRB?
Well, the second one, I mean, BOSS 100 is part of CAT 100. So that's, so that's a pretty simple question to answer. Regarding the PRBs with CAT, so you need to be careful, and I would expand this out to really any ERD technology, or let's just say anytime you're injecting any electron donor in the ground, you're going to generate daughter products, right? And in a PRB in general, depending on what your velocity and flux look like, you don't have enough residence time to be able to adequately treat everything. And so you've got a couple options. And I would say most common would be either a series of PRBs with CAT, or a CAT PRB. You'd have some gap in space. And then you'd have a BOSS 100 PRB. And so it's a similar approach, but I still think it's important. And I've actually, on some sites, now these are a little different. These are pretty low concentration polishing type sites where we've integrated CAT and BOSS in the same PRB, where we've got a couple rows of points on the upgradient side are CAT, and then a couple rows, on the downgradient side are BOSS 100. So we've got some flexibility. Really depends on the site and the conditions. But if you've got an inaccessible source and you got to put a PRB, say outside a building, and we want to look at CAT because of the concentrations, we absolutely have to look at some sort of downgradient control from there, and that'd likely be in the form of a BOSS 100.
Okay. This next question refers to both timeline and cost comparisons. And I think you covered a little bit of this in regards to, I think it was case study 1, if I recall, Mike. CAT 100 versus ERD or an ISCO application. Is there any way to compare timeline for starters, but then possibly a rough idea and magnitude of cost comparison?
I mean, I'd have to break out the specifics for that project, because that's a pretty complex question. Looking at comparing different techniques, the timelines and cost. Is that the gist of it?
It is, yeah. And I think obviously cost is very site specific. So maybe if you want to, kind of focus on rough timeline in comparison.
Right. So, if you're going to be doing, say, ISCO, so that's the kind of easier. I mean, if you're going to be doing ISCO, and let's just say it's a chlorinated ethene site, so you can use something like permanganate for it. Even permanganate, which is a pretty persistent oxidant, you're going to have to come back and do some reapplications. And so, because that's the best case if you're using something that's shorter lived, something like an activated persulfate, for example. If you've got a mixed plume, then you're looking at maybe 2 to 4 weeks of where that technology is active in general. And so you're looking at multiple mobilizations, multiple applications. All of the sites that I presented were a single application for that area. So as I mentioned, there's phased approaches, across these sites, but each of these areas were only injected once. And so I think that's an advantage of this approach, where you may not, and I think you could argue, I was going to say you don't get that as much of an immediate decrease across the board because, you know, ISCO is going to be not as, it's going to be less selective than on the, you know, bacterial side. Bacteria are going to preferentially degrade your more chlorinated species. And so you're going to get more PCE, TCE over, say, DCE vinyl, for example. So, you know, it's your day is going to look a lot different because you're going to get rebound, and which we did see on that last site, even with this. But I think in general, you got to have that kind of extreme situation and quite a lot of saturated soil mass to be able to drive that, because that's a strength of the activated carbon, is it helps wash out that rebound that you see with typically ISCO. So from a time frame standpoint, it's hard to say. You're probably, you know, like if you were going to do that first site that I showed, which had permanganate used as part of the soil mixing, you know, you're probably looking at a very similar sort of 3 to 5 year window, say. But I don't think, one thing that ISCO is not good at is achieving low concentrations. So I think one of the strengths of the technology is it can sort of handle it on the higher side, but can also deal with it from a polishing standpoint. So I think if you were going to go with an ISCO approach on any of these sites, you'd have to switch gears at some point and probably go to some sort of biological polishing anyway. And so now you're looking at 3 or 4 or 5 injections, maybe, where each of these sites was done with one.
A nice summary there. We've got 2 questions here, and then I'd encourage our attendees, again, thank you very much for spending time with us this afternoon or this morning, depending where you are. We're going to take 2 more questions, and then Mike's contact info is on the screen if you want to reach out directly to him with any additional ones. I would also note there's a couple of you that have asked some very site-specific questions regarding deployment, application, and cost. Again, please reach out to Mike or me or anyone else at AST or RPI to answer those. So Mike, the other question here was, how do I send samples to the RPI laboratory?
Yeah, so I think the best thing to do is to coordinate with one of us. But if you go to the RPI website, and there's an online chain of custody that gives some pretty detailed instructions. But I would say, given that, depending on where you're sampling soil from, sending it to RPI, there may be some sort of restrictions on, or some specifications on packaging soil specifically. If you're in, say, like the Japanese beetle zone or the fire ant zone, depending on where you're pulling samples from, you have to package your samples a little differently before they come to RPI. But I would say just, you know, get with one of us and we can coordinate that with you. Make sure we've got a good plan on the field, how many samples we want to take, and then for both groundwater and soil. And then basically, you know, RPI doesn't cover shipping or coolers or glassware, because the service is free. So as long as essentially if you get the samples to us, we'll go and we'll run them for you.
All right, and final question here. Again, combining 2 questions because I think I understand the nature of both. Can you use city water, public water to prepare this product, or do you need any special dechlorinated or anoxic water?
We have not used anoxic or dechlorinated water for any of our projects as far as I know. Now, I'm not too worried about the oxygen, because that's going to, with the presence of the electron donor, that's going to get decreased, and with the 0 valent iron, right, you're gonna knock down the ORP very quickly. I have, you know, and I've only used dechlorinated water a handful of times, and those were mainly like emulsified oil type applications a while ago. And so I think if it's a concern for a particular site, that we just get it tested and see what sort of residual chlorine concentrations we're looking at. to see, because it is a challenge to get dechlorinated water in large quantities. So depending on the site, so we don't want to use that. We don't want to specify that unless we absolutely have to.
Great. Well, again, thank you everyone for joining us. Anything we didn't address here, we tried to get to all your questions. I tried to aggregate some of them in the interest of time. If there's anything site specific, please reach out to us as well. If there's anything technology specific, you can reach us or RPI directly as well. AST's address is on the screen, and there's also links at our website to reach RPI as well. So thank you for your time, and thank you, Mike. Do you have anything else to say to wrap us up here?
No, thanks everyone for your time. Enjoy the rest of your summer.